Nighttime carbohydrate restriction is a targeted chrononutritional protocol where carbohydrate intake is completely eliminated after a specific physiological threshold, typically 4 p.m., to extend the window of low systemic glycogen availability throughout the nocturnal sleep phase. This method shifts the cellular substrate preference from glucose oxidation to lipid oxidation, lowering the respiratory quotient, RQ, and accelerating fatty acid utilization during subsequent exercise without the chronic physiological stress of continuous caloric restriction. The human metabolic architecture is fundamentally governed by circadian rhythms that dictate nutrient partitioning, insulin sensitivity, and substrate oxidation across the 24-hour cycle. In the field of sports science and metabolic longevity, maximizing fatty acid oxidation while preserving lean muscle mass is a primary objective. Historically, methods like the chronic low-carbohydrate, high-fat diet or the classic sleep-low, train-low method have been utilized to force the body to rely on lipid stores for fuel. The conventional sleep-low strategy requires athletes to perform a glycogen-depleting training session in the evening, restrict carbohydrates overnight, and perform a subsequent aerobic session the following morning in a fasted state. While this method effectively upregulates the molecular machinery responsible for lipid metabolism, the long-term implementation of such aggressive restriction induces significant psychological stress, elevates systemic cortisol levels, and compromises sleep architecture. To mitigate these adverse physiological side effects, recent clinical trials have investigated a modified protocol, the short-term intensive nighttime carbohydrate restriction method. This protocol restricts all carbohydrate intake strictly after 4 p.m. without requiring a prior evening glycogen-depleting exercise bout, seeking to achieve identical shifts in substrate oxidation with reduced homeostatic stress. To systematically evaluate the efficacy of this chrononutritional intervention, a randomized, two-group parallel design clinical trial was conducted. The cohort consisted of young adults regularly participating in athletic activities. During the baseline phase, participants maintained their habitual dietary intake to establish precise individual baselines for total caloric consumption and macronutrient distribution. In the intervention phase, both groups maintained identical total daily caloric and carbohydrate intake, ensuring that any observed physiological adaptations were driven entirely by the timing of macronutrient consumption rather than a net caloric deficit. The control group distributed their carbohydrate intake uniformly throughout the day, while the experimental group was subjected to complete carbohydrate restriction after 4 p.m. Crucially, all participants across both groups performed a mandatory 1-hour aerobic exercise protocol every morning prior to breakfast, running at a fixed intensity corresponding to 65% of their maximum heart rate. This specific exercise intensity corresponds directly to the individual crossover point, where the absolute rate of fat oxidation typically peaks before declining as intensity increases toward glycolytic pathways. The primary biochemical objective of restricting carbohydrates after 4 p.m. is to deliberately prolong the duration of low systemic glucose and insulin availability during the nocturnal fasting window. When carbohydrates are consumed late in the evening, the resulting rise in blood glucose triggers a robust secretion of insulin from pancreatic beta cells. Circulating insulin acts as a powerful molecular inhibitor of lipolysis, the breakdown of stored triacylglycerols into free fatty acids and glycerol within adipose tissue. Insulin mediates this inhibition by activating phosphodiesterase 3b, which degrades intracellular cyclic AMP, thereby preventing the phosphorylation and activation of hormone-sensitive lipase. By cutting off carbohydrate intake at 4 p.m., the experimental protocol allows circulating insulin levels to fall to basal levels hours before the onset of sleep. This sustained absence of insulin removes the molecular brakes on adipose tissue lipolysis, initiating a profound metabolic shift where the body is structurally forced to rely on fatty acid oxidation to meet its nocturnal basal metabolic demands. The prolonged absence of nocturnal carbohydrate availability induces distinct adaptations in whole-body substrate utilization, which are precisely quantified using indirect calorimetry during exercise testing. The key physiological metric utilized to evaluate this shift is the respiratory quotient, or RQ. The RQ is calculated as the ratio of the volume of carbon dioxide produced to the volume of oxygen consumed. RQ equals VCO_2/VO_2. An RQ of 1.0 indicates exclusive carbohydrate oxidation, whereas an RQ of approximately 0.70 signifies exclusive lipid oxidation. The clinical data revealed a statistically significant reduction in the resting and exercise RQ within the nighttime carbohydrate restriction group post-intervention. This pronounced drop in RQ provides definitive biochemical evidence of an accelerated reliance on lipid oxidation. Because the mitochondrial matrix is depleted of readily available glycogen stores following the extended nocturnal fast, the pyruvate dehydrogenase complex remains inactivated. Consequently, the cellular machinery shifts toward beta oxidation, breaking down long-chain fatty acids into acetyl-CoA molecules to fuel the tricarboxylic acid cycle. This accelerated rate of fat metabolism directly influences whole-body exercise performance metrics, specifically peak oxygen consumption, or VO_2 peak, and maximum work rate. Following the short-term intensive intervention, the nighttime carbohydrate restriction group demonstrated significant increases in their VO_2 peak and maximum work rate during graded exercise testing. Operating under conditions of restricted carbohydrate availability forces an upregulation of key enzymes involved in the transport and oxidation of lipids. Specifically, there is an increased transcriptional expression of carnitine palmitoyltransferase 1, or CPT1, which serves as the rate-limiting enzyme responsible for transporting long-chain fatty acyl-CoA molecules across the inner mitochondrial membrane into the matrix. By increasing CPT1 activity and upregulating the enzymes of the beta-oxidation spiral, the muscle fibers enhance their capacity to generate adenosine triphosphate, or ADP, via oxidative phosphorylation from lipid substrates, resulting in a measurable increase in mechanical work output and aerobic capacity. Simultaneously, this chrononutritional framework induces rapid adjustments in body composition, characterized by significant reductions in total body weight. However, a critical finding of the trial was that the experimental group also exhibited a concurrent drop in lean body mass alongside the loss of adipose tissue. In the context of lipid metabolism and weight reduction, this dual loss highlights a complex biochemical trade-off. When carbohydrate intake is restricted after 4 p.m., the total glycogen stores within the liver and skeletal muscle are progressively depleted to maintain normoglycemia and fuel cellular processes over the subsequent 16 hours. Because every single gram of glycogen is structurally bound to approximately 3 to 4 grams of water within the intracellular matrix, the rapid depletion of glycogen reserves results in a corresponding, mandatory excretion of intracellular water. Therefore, a significant portion of the initial reduction in lean body mass observed in short-term carbohydrate restriction protocols is driven by this rapid shift in fluid dynamics and intracellular glycogen volume, rather than an immediate degradation of structural myofibrillar proteins. To prevent actual muscle protein proteolysis during extended periods of low carbohydrate availability, the precise regulation of non-carbohydrate nutrition must be integrated into the protocol. When muscle glycogen availability is low and glycogen depletion is compounded by pre-breakfast aerobic exercise, the skeletal muscle increasingly relies on branched-chain amino acids, specifically leucine, isoleucine, and valine, as an alternative oxidative fuel source. If dietary protein intake is insufficient to meet these elevated metabolic demands, the body activates the ubiquitin proteasome pathway to break down endogenous skeletal muscle tissue to liberate free amino acids for hepatic gluconeogenesis. Therefore, to preserve structural lean mass while simultaneously exploiting the accelerated fat-burning benefits of a lower respiratory quotient, the total intake of high-quality dietary protein must be strategically optimized. Ensuring adequate protein consumption during the permitted eating window provides the necessary exogenous amino acid substrate to maintain positive nitrogen balance and stimulate the mammalian target of rapamycin complex 1, or mTORC1, pathway, safeguarding skeletal muscle architecture from catabolic degradation. Ultimately, The clinical findings regarding the short-term nighttime carbohydrate restriction method demonstrate that metabolic flexibility can be rapidly optimized without the severe psychological and physiological strain associated with long-term deprivation models. Metabolic flexibility is defined as the precise capacity of an organism to adapt its substrate oxidation to matching nutrient availability. By restricting carbohydrates specifically in the evening, the organism experiences a highly controlled, transient phase of carbohydrate depletion that mimics the evolutionary fasting structures under which human physiology evolved. This targeted depletion forces the mitochondrial network to routinely cycle between glycolytic and lipolytic pathways on a daily basis. The continuous transition prevents the development of metabolic rigidity, a pathological state characteristically observed in obesity and type 2 diabetes, where the mitochondria lose their ability to efficiently switch to lipid oxidation during fasting states. In conclusion, the nighttime carbohydrate restriction method represents an innovative, evidence-based strategy in modern metabolic medicine and longevity science. By shifting the carbohydrate window away from the nocturnal period and pairing that restriction with targeted, fasted aerobic exercise the next morning, individuals can induce rapid improvements in peak oxygen consumption, enhance their maximum work capacity, and lower their respiratory quotient toward optimized fat oxidation. This protocol effectively bypasses the chronic elevated cortisol levels and systemic stress response typical of prolonged starvation diets. However, clinical implementation requires strict attention to total macronutrient composition, specifically ensuring sufficient protein intake to protect structural tissue from fluid and catabolic shifts. By precisely controlling the timing of nutrient delivery, we can harness the body's intrinsic circadian signaling to optimize mitochondrial efficiency, accelerate fat metabolism, and expand overall health span at a cellular level.